Hello, today we are investigating a crime scene where Kanye West was murdered. We will be using DNA fingerprinting to identify the culprit. Here, Kanye was killed. We have our suspects starting with Kris Jenner as suspect 1, Kim Kardashian as suspect 2, Taylor Swift as suspect 3, and Jay-Z as suspect 4. We will identify who is the culprit out of the 4. Usually, DNA samples taken from a crime scene are too small to be analyzed, and so they go through an amplification process through PCR. Then this amplified sample is used to test as our evidence sample. We will be using VNTR from the DNA evidence as our DNA fingerprints. VNTR, also known as variable number tandem repeats, are composed of short nucleotide sequences that are repeated in DNA. These allow forensic scientists to create markers in the DNA for analysis, and we call these markers DNA fingerprints. VNTRs come in different types of alleles and are polymorphic, and the main feature we want to look at is the length of the allele for our DNA fingerprints. We use our DNA samples and run them through gel electrophoresis, where the shorter alleles move faster while the longer alleles move slower. Here is an example of a repeat. The goal of gel electrophoresis is to be able to compare the alleles that run down the gel and look for any similarities in length of the allele between the suspects and the evidence sample. We will now run our gel with the DNA samples from the crime scene, the victim, and the 4 suspects. The alleles move due to charge-charge interaction from the gel and DNA. Here's our final product after running the gel in both VNTR loci with all the alleles shown. Let us examine the first locus. We want to find any individuals that will match the sample from the crime scene. Here we can see that the victim clearly matches the sample. However, suspects 1 and 2 do not match the sample from the crime scene. Therefore, we have evidence to say that they did not leave the evidence at the crime scene. Suspects 3 and 4, however, match the sample from the crime scene. And so they are still under investigation, which is why we had to do a second VNTR locus analysis. The second VNTR locus analysis should help us narrow down even further who left the sample at the crime scene. Now we are examining the second VNTR locus analysis. Now we can see that the victim does not match the sample evidence left behind at the crime scene. Therefore, we can say that the victim did not leave behind the sample evidence at the crime scene. We already eliminated the possibility of suspects 1 and 2 from the crime scene, even though suspect 1 does match the sample at VNTR locus 2. Now if we observe suspect 3, we see that the individual does not match the sample and is excluded from being at the crime scene. Finally, we get to suspect 4 and see that this person matches the sample at the second locus as well as the first locus. With suspect 4 being a good match at both VNTR loci, we can conclude that suspect 4 was the culprit, and it was Jay-Z who murdered Kanye. It should be noted that we can also see how a parent is related to their child from suspects 1 and 2. In both loci analysis, we can see that suspect 1 and 2 both share one allele with each other, which indicates that they probably share a parent-to-child relationship. This is due to the idea that a child receives one allele from one parent and another from the other parent. This makes sense since Kris Jenner is the parent to Kim Kardashian. Here are my references.